Hello everyone, welcome back to my channel, the Biolab Scholar. So in my previous video, I told you about how to design the primers for traditional cloning. And in today's video, I'm going to tell you how to search for appropriate enzyme that you are going to use to ligate your gene in an entry vector. So I'm going to split this topic into two parts. In the first part, I will give you an overview about the importance of restriction enzymes and why searching an appropriate enzyme is important. In the second part, I'm going to actually show you how you can incorporate those enzyme sites in your gene of interest. So let's get started. So to ligate your gene into the entry vector, you have three options. So I'm going to briefly discuss each of them. So the first method is the traditional ligation method, where you amplify your gene of interest with some enzyme sites that are also present in the multiple cloning site of your entry vector. So once you, here it has shown the donor plasmid, but this can be anything. This can also be your DNA that you have directly extracted from your plant or any other bacteria or anything. And then you can use those primers with the enzyme sites to amplify the gene of interest. And then when you get the end product of that PCR, you will get your insert flanked by those enzyme sites. So you are going to digest your insert using these enzyme sites so that at the end of your digestion reaction, you will get flanking sticky ends around your insert. Similarly, you are going to digest your vector with the corresponding enzymes. Like if we have used here EcoR1 not 1, you are again going to digest your EcoR1 and not 1. And then you are going to open the vector so that you can ligate your insert into this vector. And then finally, after ligation, you are going to get your insert inside the entry vector. And this entry vector is now ready to use for further cloning. So the second type of cloning is by topo-TA cloning. So in this type of cloning, you already have 3' T overhangs in your entry vector. So what you're going to do when you amplify your gene of interest, you just have to incorporate an additional 3' A overhang in your PCR product. So when you are going to ligate both of them, they are going to ligate by this TA pairing. So that's why it is called as TA cloning. In third type of cloning, we are using homologous recombination. Here, we are going to amplify our gene of interest using the ATTB flank PCR, and then we already have a donor vector with ATTP1 and ATTP2 site that are going to recombine with this B1 and B2 site, and as a final product, your CCDB toxic gene is replaced by your gene of interest in the entry clone. And then this entry clone is now can be used for further cloning for making the expression vectors. TA cloning and BP reaction are more or less modifications of your traditional cloning. So if you understand your traditional cloning to the best, you have that leverage of modifying your protocol and have the best efficiency of generating your clones. Because so each of these techniques have their own advantages and limitations. So understanding the crux of cloning will help you in your cloning. When you will face some difficulty, it is going to help you to modify your own protocols. So I will go in details about how you choose the enzyme in your entry vector. So this is an entry vector with a magnified multiple cloning site where you can see number of enzymes. Some of the enzymes are blunt cutting enzymes. Some of the enzymes are the ones that produce sticky ends. So in cloning, it is better to produce sticky ends that provide us better ligation when we do a ligation reaction. So why do we need these many number of enzymes in our multiple cloning sites? So when we take our gene of interest sequence, there are possibility that you might find one or two or like other enzymes cutting in between your gene sequence. So if, for example, ZO1 enzyme is cutting in between your gene sequence, then you cannot, of course, use it for cloning, because if you are going to use ZO1 for your cloning, it is not only going to chop at the ends, but it is also going to chop in between your genes. And once your gene is fragmented, there is like no point of doing that cloning. So sometimes you are lucky if you can get maybe like 2 or 3 enzymes that do not cut at all in between your gene. So then you can proceed for a traditional cloning. For example, if I say that ZO1 and EcoR1 do not cut in your gene of interest. So what you can do is you can use ZO1 in your forward primer and EcoR1 in your reverse primer. And then when you amplify your gene of interest with these enzyme flanking sites, and when you digest your vector and your gene and then ligate them, there are chances that you are going to get your gene of interest in the correct orientation in your entry vector. But sometimes you are not that lucky and you only have like one enzyme option, like maybe like spare one, and then you have to incorporate only spare one in your forward and your reverse primer. And then when you do the ligation reaction, there are 50% chances that you will find your you will find your gene of interest in correct orientation and in reverse orientation. Then you have to use your PCR to find which clone is in the correct orientation. This can be a separate discussion. So, and then one more important thing to remember is that you have to use the enzymes in correct order. Like if on your multiple cloning site, you have this order, like not one, ZO1, EcoR1, spare one. And if you are going to use ZO1 and EcoR1, and you put your ZO1 on your reverse primer and EcoR1 on your forward primer, you are of course going to get a reverse orientation of your gene in your entry vector. So it is important to remember that you have to be careful about which enzyme site you are putting on which side of your primer. And then once you have your insert and vector cut with the same enzymes, like here ZO1 and PST1, you are going to ligate them. This is a correct ligation reaction. But if you see in this example, if you cut your insert with ZO1 and PST1 and you cut your vector by SAL1 and PST1, the ligation is not going to happen because you do not have the corresponding sites of ZO1 in your vector. So where your ZO1 can stick together, because these enzymes are the ones that produce sticky ends, and they need the other half to be stuck together. So if you do not cut your vector and your insert with the correct corresponding enzymes, your reaction is not going to work. You are never going to get your correct product. So you have to be very careful about what enzymes to pick and what order of enzymes need to be incorporated in your forward and in your reverse priming. So yes, that's all for today. I hope you find the video helpful. And if you have any questions, please comment down below. I will try to help you as much as I can. And if you like the video, please do subscribe, share, and comment. I will really appreciate it. And in my next video, I'm going to talk about how you actually incorporate your sites and how to find the enzyme sites in your gene of interest that do not cut in your gene of interest. So yes, stay tuned. And till then, stay healthy, stay happy, and see you in the next video. Bye-bye.